Hey, okay, so we are going to start looking at how do we do well, how do we pass, or even how do we get an A in our biology class in this series of videos. And this portion, we're going to talk about how we can use the textbook. So this video goes along with a sample lecture about viruses, and this sample lecture is based on or associated with, assuming students are going to do reading in the textbook chapter going along with that. And this textbook is called Biology. It's produced by OpenStax, and that is a free online textbook. So there's a link to that with this video. And so you don't have to buy a printed copy. I'm old school. I'm used to having a printed copy of a book. It's about $50, but it's free online. So when you're looking at your book online, you can basically use the same techniques as I'm going to show you on paper. So it's just easier for me personally to be looking through the paper book. All right, so I'm going to assume that you haven't actually had the lecture yet or looked at the lecture slides, and we're going to do a really preliminary review of the textbook. So when you have time, this is the very best approach. Now, if you don't have time and you're only going to come to the textbook after you have listened to the lecture, that's okay too. It can save you a little time, but here's a great way to get started. All right, so here's the chapter that's assigned. The chapter outline has 4 sections. The first one's on viral evolution, morphology, and classification. The second section is on virus infections and hosts. The third, prevention and treatment of viral infections. And then 21.4, the fourth section, other acellular entities, prions, and virions. If you do look at the lecture beforehand or you have instructions from your instructor, then for this chapter, I let you know that I'm not going to cover that last section, 21.4, so you can skip it. So that is going to save you some time if you get instructions from your instructor ahead of time of which sections of the textbook you're going to use or not use. All right, so to familiarize you with what's in this chapter, this little introduction, you shouldn't try and take notes. Usually on the first little section here, that's kind of to give you an overview. Everything that's in there is going to be gone over in more detail later. The beginning of each section, 21.1, it gives you, by the end of the section, you'll be able to do the following. For example, describe how viruses were first discovered and how they were detected. All right, so after you read each section, you should go back to this list of learning outcomes and actually try and write out or draw the answers or the results of these different outcomes. All right, so the chapter is going to start off, viruses are diverse entities. Great. They vary in structure, method of replication, and the host they infect. All right, so those are examples of how they're diverse. All right, so when we're first reading the textbook chapter, we might only want to read the first little paragraph in each section. That gives us a general introduction to what the topic is. A couple other things you want to notice. We've got some words that are in bold. All of those words that are in bold are going to be defined. We go to the end of the chapter. There's key terms, and so we're going to have a written out in words definition of all of those terms that are in bold. Those are important. Those are ones that you should know what they mean in the context of our chapter, right? Because if I gave you an exam question and it had the word envelope in it, and you're thinking, well, that's a folded over thing that I'm going to stick a stamp on and send in the mail, right? Or you look it up in your Google, that's not going to be a good answer in the context of this class, because here the envelope is specifically a lipid bilayer that encircles some viruses. So it's an envelope in that it surrounds the virus, but it has a specific meaning in the context of our chapter. So at the end of each chapter, you're going to have your list of key terms, or this is sometimes called a glossary. And then after that, there's going to be these paragraph chapter summaries, some practice questions, review questions. There's multiple choice ones. There's critical thinking questions. And in this textbook, all the answers, where'd my page marker go? All the answers to the questions are in the back of the book. So as you're going through, you can practice doing reading comprehension, and you can find out whether you answered that question correctly or not. So that's something you want to know about. All right, so as we are going through our very first preliminary reading of our chapter, we want to just note these words in bold, right? Tobacco mosaic disease. That's an example of a virus that infects plants, and specifically tobacco plant. And so you want to just read enough of each of these sections that you can get an idea of what those words in bold mean. So virions, a single virus particle. Right, viruses aren't cells, they're acellular, so they don't have a cell membrane, they don't have ribosomes, so we got to call them something. We call them virions or particles. All right, you also want to look at the figures or images, and this is the virus here in this picture. So you want to identify what is in the image. And notice there's scale bars in these images. The point of this picture is to show you how tiny viruses are. These are scanning electron micrographs. They're not light microscope images. Or they're transmissions, sorry. So that's in the legend to the figures here. And if we read this, we see, okay, this is the virus particle. This happens to be bacteriophage. They look like little spaceships. It's sitting on the surface of a bacteria. And then this picture over here is showing bacteria relative to a eukaryotic cell. So sort of as small as bacteria are relative to a eukaryotic cell, viruses are that much smaller relative to the bacterial cells. So the purpose of this image is to give you a sense of scale. Right, so the evolution of viruses, there's some things in italics here. So that calls out kind of, oh, there's a couple different hypotheses here. Our preliminary reading of the textbook, don't worry about what those hypotheses are. Now we get to viral morphology. Now make sure when you're reading these titles, you know what all the words mean to start with. Right, morphology is just the shape, right? What do they look like? What's their structure? Now we have some vocabulary words here, right? Viruses are non-cellular. That means they are biological entities that do not have a cellular structure. It's really important that you understand what that means. So it's being mentioned over and over again in this section. So make sure that you have kind of wrapped your head around that. They have this protein coating called a capsid. And then sometimes an outer envelope outside of that. All right, so capsid and then envelope. There are, so the capsid is made up of capsomeres, and each of the proteins of the capsid are encoded in the viral genome. All right, so we know that DNA is used as instructions to make proteins. So the fact that there are these little protein subunits that make up the viral capsid, we need to have genes that code for those proteins. And each of the individual proteins is called a capsomere, and they get assembled together into the overall capsid. So it's kind of like, if you think about actin and myosin, those are proteins, and they're assembled together into those thick and thin filaments in the muscle cells. So we're sort of making things out of subunits like we do in biology. Now I put an arrow here next to this figure. If I did peek at the lecture ahead of time, I know that I have a very similar figure in the lecture slideshow. So that's when we might want to make sure we read this section more carefully our second time through, because it's something the instructor has called out as important. It isn't this exact same figure, it's a very similar one, but your instructor might know of or have found one that they think is better than this figure, and that's why they would put that one in the lecture instead. All right, so we have three different kinds of virus particles: helical, icosahedral, and complex. You don't need to worry too much about what those look like. It looks like here, helical is sort of like a spiral or a tube. Icosahedral is more geometric, and complex is sort of anything else. All right. So the next part of this section, we're still looking at morphology. And then we have the next vocabulary word is viral receptors. So the receptor is the place where the virus attaches to the host cell. And in this figure, there's a lot of stuff going on, but we see that there is this region where the CD4 protein is fastening to the HIV virus. In this case, it's got these little green things sticking up. And so this is an important concept. Now, you may be thinking, oh my gosh, I'm going to have to memorize that HIV has this GP120 protein sticking up and then it binds to the CD4 molecule. Don't assume that yet. It depends on your instructor. It depends on what class this is. Don't sort of get frightened about all the things that you might have to learn at this point. Just keep kind of going through the chapter and saying to yourself, what's the first most important concept that this figure is trying to show? Well, it's an example of this viral receptor, right? The virus has to bind to the host cell. That's incredibly, incredibly important. All right, on our next page, we have, again, some more examples of complex viruses. These are different examples. There's some other vocabulary here. Again, this is a figure or a similar figure that the instructor used, and there's a little practice question here. So definitely as you're going through the chapter, if you have read this carefully, you want to go through and do the practice questions because that's a form of active learning. Active learning is where you participate in the information rather than just casting your eyeballs across it. So you want to think about what you have been learning. All right, our next topic is types of nucleic acid. Make sure you're thinking about what is a nucleic acid, right? We've got deoxyribonucleic acid and ribonucleic acid, which we also call DNA and RNA. And in eukaryotes, we only have DNA as a genetic material, but viruses can use either DNA or RNA, right? That's interesting. That's something you want to make sure and remark on. And then when we look at the next section, virus classification, there's challenges. Okay, viruses are really diverse. It said that in the very first paragraph. And in biology, we always want to classify things. How can we look at all the different kinds of viruses and talk about what kinds there are? Well, we can categorize them different ways. That's the purpose of this section. So when we look at viruses, they could be DNA or RNA, they could be single-stranded or double-stranded. In eukaryotes, we have double-stranded DNA. That's the only thing that is used for genetic material. But we can have single-stranded or double-stranded in viruses. All right, something weird they do. It can be linear or circular. Well, chromosomes that are linear look like so, and that are circular look like so. All right, now, previously you may have learned that eukaryotes have linear chromosomes and prokaryotes have circular chromosomes. Turns out viruses can have either. All right, then we can have non-segmented. Genome consists of a single segment. Thanks. Makes more sense when you look at segmented. Genome is divided into multiple segments. All right, what does this mean, segments? Well, it's similar to how we have for eukaryotic organisms, the genetic material divided up onto different chromosomes. So these segments are equivalent to chromosomes. They're just separate pieces of the genetic material. All right, looking at our next type of classification, we can have naked icosahedral, enveloped icosahedral, enveloped helical, naked helical, blah, blah, blah. What's the difference here? Well, we've taken these categories that we were defined, helical, icosahedral, complex, and we've added another layer to what kinds we can have. And basically it's saying, well, they can either be naked or they can be enveloped. They can have that little bit of cell membrane wrapped around them. Right, now notice there's also lists of all these examples in the table. Don't freak out about having to memorize all of those. It just depends on your instructor. I personally would not ask you to be able to tell me what capsid type the mumps virus has, right? That's just me. You might have an instructor, if this were a virology class, you might need to memorize all of that stuff, right? But don't freak out about it yet. All right, now we get to this Baltimore classification. The Baltimore classification is what is sort of currently used. It's the most formal and systematic type of classification. And so these groups, these different groups and their characteristics, this is covered in the lecture. So we're going to come back to this. This is going to take a little work to dive into. And we're just going to put a sort of a check mark in our mind to come back and figure out what this Baltimore classification system means. All right, so that's section 1. That should have taken maybe 5 or 10 minutes in your first reading. So our next section, virus infections and hosts, we have another little list of things we should be able to do by the end of reading the chapter. And it starts out by saying viruses are obligate intracellular parasites, right? Pause there. Obligate means they have to do it, right? Intracellular means they're inside the cell, and parasites means that they exist in this symbiotic relationship. Remember, it's not a mutualism, it's like a parasite. So like a mosquito is a parasite, it takes something from the host. And so viruses are required to have a host. They can't live without them. All right. Now this paragraph talks about this idea of permissive. That means that every type of virus only has a certain type of cell that it can infect. And that has to do with the specificity of that receptor that it's going to have on its surface that's going to bind to some part of the cell that it's infecting. So that's the basic idea of this paragraph. All right, now we have steps of virus infection. And the idea here is that what does the virus have to do in order to access that host cell? And then how does it proliferate? And then how does it get out of the cell to spread infection to other cells? So this breaks it down into 4 steps. We've got attachment, we've got entry, we've got replication and assembly, and egress, getting out, right? So this is going to give some examples of the ways that viruses do these different things in this section, and it's going to go into more detail in the next few pages, because it turns out that different types of viruses, the kinds of organisms that are infected by them, these things happen differently. So many things in biology are context specific, right? When we're talking about eukaryotes versus prokaryotes, things are going to happen differently in them. So we have to make an important distinction and be clear in our mind what we're talking about. And so notice that we have this picture here is a figure that's going to be used in the lecture. I've marked it here, so we want to go through it in more detail and understand it later. This is influenza. So this infects humans, so we know that this is infecting animals. All right, so we're going to have viruses that infect eukaryotic organisms, and if we sort of scan ahead, we'll see that we have a distinction made between plant viruses and animal viruses. And we're going to start with prokaryotic cells here, right? So we want to look at how do these 4 things happen in prokaryotic cells that are being infected by viruses, animal cells that are being infected by viruses, and plant cells that are being infected by viruses, right? And thinking about how those cells are different than one another can help us prepare our minds to thinking about what's an important difference between how this might happen. For example, prokaryotes have a cell wall. Plants also have a cell wall. Animals don't have a cell wall. So viruses might have an easier or harder time getting into cells because of whether they have a cell wall or not. All right, so we're going to look at this figure. We see that there's a step of attachment, entry, replication and assembly, and then egress, right? So we're being shown for an animal the different steps in the way that they can happen. Then we have this second page, this figure, also one that's shown in the lecture. This is for bacteriophage, right? Phage infects bacteria. So these are prokaryotic. And it turns out there's two different things they can do, the lytic cycle and the lysogenic cycle. And we're just going to make a note to start with. We've got these two different things that we need to figure out which is which. Lytic, lysogenic, they both sound kind of similar. Lysis is the breaking or the exploding or the disintegrating of a cell. So they are going to have something to do with that. We're going to see in this figure this little bacteriophage guy on the outside, right? So lytic cycle, lysogenic cycle, that has to do with bacteria and with bacteriophage. For ask a question about animal cells, there is no such thing as lytic and lysogenic cycle. All right, next thing we're going to look at plant viruses. How are they different? And the thing that's interesting here is plant virus, to enter a new host, it has to have some kind of mechanical damage. Right, so just a living, healthy, intact plant cell can't get infected by a virus. That's pretty neat for plants. And we have some bold words here: horizontal transmission, vertical transmission. Transmission is how the virus gets from one host to another. And so let's just think for a minute about horizontal transmission. When we think about the horizon, that's a straight line. So we might be thinking transmission is going to go from side to side, and vertical transmission is going to be up and down, right? So we're going to have transmission going from up and down, right? Virus from a parent, all right? So from mom to offspring. And then horizontal is going to be one plant to another, like your next door neighbor, all right? All right, animal viruses, there's some more information here. Acute, chronic, asymptomatic infection. Now, since we've been living through COVID for the last couple of years, we know that some people can get infected by the virus and be asymptomatic. They don't feel sick, all right? So they're still carrying the virus. They can pass it on to somebody else. Acute disease is when you get really sick, your symptoms are worse, you might require hospitalization. And chronic infections, different kinds of diseases do this and others don't. So hepatitis, herpes, those are things that do chronic infections, and the symptoms, like they never get eliminated from your body. They can pop up every once in a while when you're under a lot of stress, for example, or sick from something else. So those are the definitions of those words. Now, you probably know these already, so don't freak out. Don't get to a place where you know less than you did before you started reading this book. We're just putting very specific words to those ideas. All right, and then we have a neat thing here, oncogenic viruses, right? So somebody who studies cancer as an oncologist or treats cancer, oncogenic viruses are viruses that cause cancer, right? That's pretty scary. All right, the last section that we need to review for assignment is prevention and treatment of viral infections. We have gotten a lot of information about this over the last couple of years in this section. So we know all about PPE, right? Personal protective equipment. We know about vaccines, antiviral drugs. So this section might trigger some emotional content for you, but it might also be super duper interesting, all right? So we're going to identify major viral illnesses that affect humans, compare vaccinations and antiviral drugs as medical approaches. All right, so this figure up here, now this figure is going to be in your slideshow. And again, don't freak out that you're going to have to memorize all these different viruses. And there's some crazy ones on here. I've never heard of Coxsackie A, for example, kind of interesting. So wait and see how this material is covered in your lecture. I can give you a preview. I can tell you that what I talk about in my lecture is viruses can cause a variety of different kinds of symptoms depending on what tissue they infect. For example, a virus that infects your lungs may cause pneumonia. All right, so that's the level of detail I'm going to cover this information, right? No freak out necessary. Don't start making flashcards of these all these different viruses. All right, next section about vaccines. What do they do? How are they made? Now, this material, you kind of have to know some things about the immune system in order to really understand this. So personally, I'm putting off talking about a lot more about vaccines until we've talked about the immune system later on in the semester. All right, so look at some of these. What's the idea of a vaccine? Kind of read this. There's not a ton of content there. And again, giving a vaccination in order to treat rabies is an example. These are just kind of examples. There's actually not a ton of content here. All right, so notice there's no more bold words, just the idea of what an antiviral drug is. It's a drug that somehow is able to treat a virus, right? How come we can't use an antibiotic to treat a viral infection? Well, antibiotics attack cells, right? And so viruses aren't cells. So that's an important take-home message about this section. All right, we have this incredibly complicated figure about HIV. It gives me a headache just looking at it. I'm going to skip it for now. And now we get to the last section, 21.4. Right, my professor told me I didn't need to read this, but piranhas are cool. I want to know what they are. What the heck is a viroid? I don't know. All right, so feel free to read this section if you're interested. It's a couple paragraphs. It's not that much. These other sections, these yellow sections, these are kind of neat, kind of extra stuff. If your instructor covers this material in lecture, you might want to read this more carefully, but you want to hold off on that. Oh, here's a virologist, a person who studies viruses. So if you're interested in that. Oh, American Society of Virology website here. So if you haven't decided on your career path, if you might be interested in maybe not being a doctor, but doing something studying diseases, then there might be some resources here. All right, so that's your preliminary trip through this chapter. It wasn't too traumatic. You have prepared your mind for further learning. We haven't even taken any notes yet, and I'll come back in a few minutes and we'll talk about what notes you should have and how they're going to interact with your notes from lecture, because that's an important thing to keep in mind as well.